Good morning, good afternoon, and good evening to everyone that's joining us from around the world. We're so glad that you could be here. My name is Austin Bailey, and I'm a representative with Chiral Technologies. Today's webinar will be presented by Dr. Weston Umstead, and will cover the most versatile and widely applied technique for enantiomeric separation, chiral chromatography. Wes is our senior product manager and has been with Chiral Technologies since 2016 and oversees our free in-house method development screening, as well as our custom separation services. Wes has been published in numerous application notes and journal articles and hopes to share his experience and knowledge here today. As we proceed, please feel free to post any questions that you may have in the chat, and we'll be sure to take 10 minutes after the presentation to address those questions. Without further ado, I present to you Dr. Weston Umstead.
Thank you, Austin. And again, thank you to everyone for joining this morning, this afternoon, this evening, wherever you're located at. The focus of the conversation today is going to be on chiral chromatography. We're going to do a little bit of a deep dive into some of the fundamentals and the mechanisms. We're going to start with a discussion around, just a refresher. We want to make sure everyone is familiar with chromatographic terms before we really launch into a deep dive and discussion on chiral chromatography specifically. We'll talk about some of the major classes of selectors that are available. Obviously, if you know dicell chiral chromatography products are primarily polysaccharide-based columns, but there are many other options, so we'll cover what those are. An introduction to our phases and talking a little bit about the separations mechanism that is unique to the polysaccharide phases. A very brief little applications and examples. To be honest, we could probably do a full 24-hour webinar just on the applications alone, and we obviously don't have that. So just kind of summarizing some of the major classes and separations that we have, and then finishing with some closing remarks and good information for you as you leave this webinar today to follow up on your study of chiral chromatography and some services that we have to offer that might be important or relevant to you. So let's start with that refresher, the importance of chiral chromatography. What is chromatography? Very, very basic, simple concept definition. It is a separation technique based on different interactions of analytes with a stationary phase, that's the thing that's not moving, and then the mobile phase, the thing that is moving. The example that I have shown here is just a very simple paper chromatography. You've probably seen or even done this experiment yourself. You blot a mix of ink colors in one spot, and then over time, as the mobile phase is allowed to travel across the stationary phase, you see the different colors of ink beginning to separate. Most chromatography is utilizing some physical or chemical difference between the analytes and the stationary phase in order to make the separation happen. And as we start talking about chiral chromatography, that changes a little bit because there are no real physical or chemical differences between them to try to harness to make a separation. Now, we talk about chirality. Again, just very briefly, if we were to talk about the textbook definition, we're talking about non-superimposable mirror images of each other. Most often, we will talk about hands, left hand and right hand. But chirality happens in nature, not frequently, but there are examples of it. You might be familiar with tartaric acid crystals and separating those by hand. Louis Pasteur did that back in the 1800s. But even naturally occurring things like shells, sometimes you'll see those are chiral to each other, or the formation of different cacti are either left-handed or a right-handed spiral. But we see it in DNA as well. You've got bDNA and zDNA, which are left and right-handed forms of a double-stranded helix for DNA. So chirality happens in nature sometimes, and it can have some interesting effects, which is what we'll talk about as we move through this presentation. Now, specifically, we talk about chirality, we're often going to be talking about enantiomers. So enantiomers here, again, non-superimposable mirror images of each other, but it's two compounds that have the same functional groups that vary only by the orientation of those functional groups in space. Now, in this example, I have here an actual chiral center. So you'll see chiral center or stereo center be used quite frequently, but it could also be things like axis of symmetry as we start talking about things like atropisomers. Atropisomers are not, they can have a chiral center in them, but there is an axis of rotation that allows for two different distinct conformations to exist at any one time, and those can also be treated as if they were different things as well. Non-superimposable mirror images here, left hand to right hand. Obviously, if you try to superimpose these on top of each other, they would not overlap completely 100%. So why is chirality important? Again, just kind of covering some of the very basic things here before we start our conversation about more heavy topics like the separations mechanism. Obviously, biological systems are chiral. Most of the things that we see in nature, proteins and enzymes and DNA, are all three-dimensional, and they all can oftentimes distinguish between enantiomers differently. If you introduce a particular mixture of compounds, they may or may not bind differently or result in a different effect, whether it's the R or the S enantiomer. We can talk about things like different effects. If one might be a therapeutic, the left hand might be therapeutic, the right hand may be inactive or maybe even harmful, which leads us to the very classic example of thalidomide. It was a morning sickness drug that was prescribed in the late 1950s, early 1960s. The unfortunate reality was that the R enantiomer gave the therapeutic benefit of relief from morning sickness, but the S enantiomer led to a number of really terrible birth defects in the children that were born from the mothers that took this. So it was a very potent teratogen. Now, you may see thalidomide popping up nowadays being used as a cancer drug. It's used in a number of cases for things like Protacs as a sort of a head unit. It's sort of the detonator head unit that is used in those Protacs. So there are some uses for this drug, but in its original iteration, it was a pretty classic but terrible case of why chirality is important and why it needs to be accounted for. Drug development and regulation. So because of things like thalidomide, you had regulatory agencies like the FDA that stepped in and said, hey, if you're going to make these compounds and sell them as a mixture of enantiomers, you need to be able to characterize and ensure that each one is safe on its own, or that each one doesn't, cause these sorts of effects that you're seeing with thalidomide. So in response to thalidomide, but also some other cases, the FDA launched in 1992 its policy on chiral drugs, which required that chiral purity be reported in all cases where there is chirality within that molecule. And it emphasized that if there is no potency for one of those, or obviously if one of them is unsafe, that the single enantiomer must be produced for that drug when it is launched to market or pass through clinical trials. So an emphasis on single enantiomers at this point became really important. Of course, there's other things like chemistry and materials. Chirality can influence things like catalysis and asymmetric synthesis. So we're not just talking about single small drug molecules. We can talk about really large macromolecules as well. Chirality can be a major influencer in those systems. So let's put all these different basic definitions together: chromatography, enantiomers, chirality, and specifically say, what is chiral chromatography? It is a separations technique based on different interactions of analytes with a chiral stationary phase and a mobile phase, which you might be thinking to yourself, boy, that sounds exactly like the definition you gave me for chromatography, and it is. The only subtle difference here is now I've said chiral stationary phase instead of just stationary phase, because we have to talk about a stationary phase that is emphasizing or using different things other than just differences in, let's say, polarity. Geometric orientation in space and chemical properties are the same, so we have to modify our stationary phase to now interact or exploit the difference in chirality that we're seeing in these molecules. Of course, chiral chromatography is oftentimes done in HPLC mode. So you see here, you've got a kind of a cartoonish version of a column where you'd make an injection of two enantiomers RNS together as a nice purple slug. And as it begins moving down the column, you'll start to see it separate into its distinct colors of red and blue until eventually it reaches the end. And then we have our nice chiral separation. The anatomy of an HPLC, we can just talk about this very briefly as well. Of course, for our topic of conversation today, the column is going to be the most important element, but there are a lot of variations on what an HPLC system can look like now. Obviously, the basic components that are going to be things like a degasser, a pump, an autosampler, some sort of detection mode, and then of course the column. Single column, a column oven might be a single column. It could be with a column switching valve. As you see here, this has the ability to screen 12 columns, but 6, 8 for a lot of different varieties of systems available from a number of different manufacturers. Now the detectors here, UV is probably still the most relevant, but mass spec detection, even chiral detectors, things like PDA or PDR detectors, sorry, CD detection. There's a number of other options that we have available to us now. But the basic principle is here. You've got to have something that pumps your mobile phase, your column, and then a way to detect it. And now we could talk about some of the definitions for chromatography as well, especially when we start talking about the separations mechanism. We start seeing things like retention factor and selectivity and resolution, and how do we start playing with those terms when it comes to our chiral separation. But we have to talk about what they mean first before we can really start talking about how do we affect them. So first one we'll talk about is retention factor, which is K. You might also see it referenced as capacity factor, K prime. They really are describing the exact same thing, which is how well is a peak being retained relative to the solvent front or to the column void. The way that we define that is by K. TR is the retention time of the actual compound. T0 is either the solvent front distance or the retention, the void time for the column when we're talking about chiral HPLC chromatography. So it's the ratio of those TR minus T0 over T0. And so that gives us our K prime factor, describes how well is that column retaining that particular enantiomer or compound. The other one that we're going to see used quite frequently is theoretical plates. That's large N. It's our description of column efficiency, or how sharp are the peaks. There are a number of different ways to calculate N values, and it depends really on some cases the system. There are different groups that calculate it differently. So you'll see it represented in a variety of different ways, but these two equations here are the two most common. Again, here when we see TR, we're talking about the retention time of the particular analyte, and then W is going to be the peak width, and that's really where a lot of the variability comes in. Peak width could be assessed either at half the peak width height, or for sigma. There's a lot of different variability, and each manufacturer and group has a little bit of a preference on how it does that. And then the last one that we're going to see often is selectivity, alpha. It's basically telling us how well are the two peaks separated from each other. Alpha is probably the most straightforward one to calculate. It's the ratio of the capacity factors. So K2 over K1 gives us the selectivity. Now, there are some inter-use of terms. Oftentimes, selectivity and resolution are oftentimes interchangeable or interchangeably used. But as you see here, they are actually different from each other. So we'll try to make that distinction as we talk about optimizing our separations. Selectivity is a reference of how well are the two peaks separated from each other. Resolution is really the definition as a whole of how good is the separation, including factors like theoretical plate count and retention factor. So the one equation that you'll see used most often is listed here. RS is the resolution. Then you've got your theoretical plate value, your retention capacity value, and your selectivity values as a way to feed into the overall calculation of resolution. So if we look at how that applies to an actual chromatogram here, your capacity factor, again, or your retention factor is describing how well is a peak retained in the actual separation. Selectivity is how well are two peaks resolved from each other, and then peak height or the efficiency in plate count is determined based on the peak height and then the ratio of that. So we start looking at how do we play around with our column chromatography, our separations. We look at the column efficiency. Again, that's our plate count number. Column efficiency is affected primarily by the efficiency of column packing, which can be things like particle size. It can be things like column length. It can be flow rate affect our plate count. So that's a parameter that we oftentimes aren't looking at when we're talking about optimizing or modifying our separations, primarily because unless you're the one actually packing the column, you don't have much control over the plate count of that separation or the plate count of that column. Of course, plate count changes depending on the actual method that you're using it in. So for all intents and purposes, it's not something that we always have a really good handle on on controlling when it comes to optimizing our separations. But retentivity, or the capacity factor, is definitely something that we have control over. It's affected most by the stationary phase and the mobile phase composition. So the more combinations that we have available to us, the more we can kind of play around in that landscape of, polar, non-polar, different column chemistries. We can have a lot of swing and effect on our retentivity. Then interestingly enough, our selectivity is also affected by a lot of those same parameters as well. So if we're talking about changing our separations, retention and selectivity are the two that we're going to be most interested in being able to modify or affect to change our separation. So we talked about chirality. Let's talk about achiral chromatography first. So reverse phase mode separations, the things that most of us are probably very familiar with. Now, when I say achiral, I'm talking about anything that's not a non-superimposable mirror image. So it could be diastereomers, it could be regioisomers, it could be structural isomers, anything that's not enantiomer or chiral, we can talk about an achiral separation for. With those, most of the cases are going to be using differences between physical and chemical properties in order to make the separation. Like I said at the very beginning, maybe it's difference in polarity between the analytes from each other, or the difference in polarity between the analyte and the stationary phase. But we have some reasonable, easy to play around with handles to change our achiral separations. Again, differences in analyte and phase chemistry is oftentimes what we're using, and it's polarity. So we have all these different achiral stationary phases, things like C18, which is more in our non-polar region, but we also have things like cyano and silica columns, which are more in our polar region. So if we screen any mixture of achiral molecules, as I have shown here, usually what we're going to see is a pretty predictable order of most non-polar, let's say, things being more retained on our non-polar stationary phases. So if we're using a C18 and we have toluene should be more retained than our phenethanol or our parafluorophenethanol, which are far more polar. They are coming out earlier. Of course, if we reverse this and we did this on a silica column, you'd see that toluene was coming off first, and then the phenethanol two compounds would be coming off last. And we can even do things like changing around the overall composition of the mobile phase and still get elution orders and retention times that are almost roughly the same. But now as we start transitioning into our chiral chromatography, all of this stuff kind of goes out the window. So the predictability becomes much less reliable. And of course, like we talked about, the separations mechanism needs to be changed because we can't just use polarity anymore. So take these into consideration as we start this journey. Achiral chromatography, not saying that it's easy, but it's easier, at least in terms of a prediction and what's going to work for these separations. So with that, we'll start talking now about our major classes of chiral selectors. And as I mentioned, if you're familiar with diacell chiral technologies, the polysaccharides are definitely one of the most widely used phases. It's what we are the leader in the market for. But there are a lot of other options as well. And a lot of the development in history can kind of be traced back to the 1970s, 1980s, where we saw things like the Perkel phases start to show up, the protein-based columns, cyclodextrins, crown ethers. In the 90s, though, and 2000s, there have been other innovations. Primarily, we've started to see things like immobilization starting to become more readily available in the crown ethers first, but then from diacell with the polysaccharides in the 2000s. We've also seen some specialty selectors showing up, things for anion exchange and zwitterionic chiral separations. So let's start our journey back with the ligand exchange phases, which are kind of one of the first examples of chiral phases that were developed in the early 1970s. The phases were originally designed to separate things like alpha amino acids, alpha hydroxy acids, and other Lewis-based molecules. So they're a little bit limited in terms of their overall applicability. They're certainly not a universal selector in by any means. They require something like 2 donor atoms, a nitrogen or an oxygen, within a proximity to each other that can form a 5-member transition state with a metal ion. So they're a little bit limited in their applicability. Their mechanism, as I said, does involve the use of some sort of a metal ion. It's typically copper, and you form these ternary complexes like I have shown down here, where it comes in, it interacts, and then it can depart in that metal complex. The mobile phases are a bit complicated. They do require the use of metal salts, which means that at least for mass spec compatibility, it's not there. good for UV detection, and certainly at the time for challenging compounds like these amino acids, it was definitely a great innovation. But since then, we've come up with some improvements that allow for a little bit more versatility and utility for amino acids, especially. The one that, of course, NiCel is most famous for, the polysaccharide-based phases, principally developed and investigated by Yoshio Okamoto, shown down here, are based on either carbamate or benzoate small molecule selectors used to derivatize polysaccharides like amylose and cellulose. Those are the two biggest ones, but also things like chitin have been used in some literature as well. The nice thing about these phases is that once they are immobilized or coated onto a silica gel support, which gives it a lot of robustness, they offer a very broad recognition domain, so not limited to just the alpha amino acids or Lewis base molecules like the ligand exchange columns are. They really are applicable to a very wide range of different chiral molecules. Of course, you know, from us, we've got a lot of chiral phases available. There's a lot of different functionality that's possible. They are also able to be used in both analytical and preparative scale separations. They have a high loading capacity, so being able to accommodate both types of analyses or separations is something that the polysaccharides offer. And especially when we start talking about things like immobilization, compatible with a lot of different solvents and detection modes. So this was, the polysaccharides really were kind of the first big leap forward in terms of universality when it comes to chiral separations. But we also have things like protein-based chiral stationary phases. We've got AGP, HSA, CBH. They're all based on naturally occurring proteins that can be isolated from nature. When immobilized onto silica, much like the polysaccharides, they do offer quite a broad recognition domain. They are, in many cases, could be considered almost a universal selector. They work on a really wide range of different things. And again, there's plenty of different proteins, so your imagination is really your only limitation here. There's a lot of different functionalities that are possible. They do, however, have a limited loading capacity because the separations mechanism is based on the actual protein binding to the analyte that's coming in. As long as there are binding sites available, the column works great. But if all the binding sites are blocked by these interactions, then you can really quickly overload the protein. And because they can be denatured quite easily by changes in pH or certain solvent exposure, they are really limited to to reverse phase mode, which makes optimization a little bit more challenging with these. We also have things like cyclodextrin-based CSPs developed by gentlemen like Dan Armstrong. You have the general structure shown here. This is not a universal structure that you see here, but they do offer a broad recognition domain, much like the two phases we saw previously. But because they require they use an inclusion mechanism, they are limited by the size. So you'll see things like alpha, beta, and gamma cyclodextrins. The overall size of the cyclodextrin is what is a limiting factor. So if you've got a really big chiral molecule that doesn't fit in to that inclusion mechanism, then they might not work so well. There are multiple functionalities available. If you notice on the outside of this cyclodextrin here, there's a lot of R groups, so we can modify those R groups and put a lot of different pendants on there. Because it's an inclusion mechanism, they do suffer from loading issues, so it's mainly an analytical tool, but they are useful in a very wide range of mobile phases as well, including GC. The Perkel type phases from William Perkel, they are, again, this is just sort of a general structure. They are considered a very wide, a broad recognition domain as well, just like some of these other options. They can be heavily derivatized and heavily modified because you've got these different functionality that can be installed here, but also the overall shape of this piece of the selector. A lot of different variety can be installed with these phases. You might also see them being referred to as brush-like chiral stationary phases, but Perkel, brush-like, it's the same exact thing. They are useful in a very wide range of mobile phases as well, but sometimes struggle when the amount of water increases above 50%. So, but they have become very popular as well. Things like Welco 1 is a very popular phase still to this day. So very brief run history through the early development phases. By the end of the 1980s, we had a lot of good options, is basically what those slides are telling us. We had things like protein phases and polysaccharides and cyclodextrins, a lot of good options available, but there's still some gaps that were there at that time. Some of the challenges included things like the separation of strong acids. especially when there was not very good functionality in order to interact with or give good UV signals. Challenges in scaling to prep. Well, we did have the polysaccharides, which have really good loading capacity. If we have issues with solubility with those coated polysaccharides, which came out first, we're limited to alcohols and normal phase mode, primarily reverse phase mode to a degree, but we can't use DCM, we can't use ethyl acetate. Detection challenges. Most of those things like ligand exchange columns could not be used with mass spec mode. So a little bit of a limitation, especially as we start looking at more complex chiral molecules like we have now. And then again, some limitations in terms of solubility of the actual phases themselves not being able to be used with water or acetonitrile or DCM or ethyl acetate. So as we moved into the 90s and then into the early 2000s, we had gentlemen like Wolfgang Lindner who developed the alkaloid-based chiral stationary phases to address, in this case, the strong acid problem. Here you've got a quinine or quinidine-based alkaloid selector, which can separate really well things like free chiral acids, lactic acid, or protected amino acids, FMOC protected amino acids, or chiral sulfonic or phosphoric acids, all mass spec compatible mobile phases as well. So kind of solving two problems here at once with these alkaloid-based phases. For zwitterionic molecules like acids, amino acids, whether they be alpha, beta, or gamma amino acids, Zwix plus and Zwix minus work incredibly well for those, as well as other amphoteric and zwitterionic molecules, which can be challenging to separate on polysaccharide or other phases because of the diversity of interactions that are taking place, the diversity of functionality that those molecules have. Also mass spec compatible, which is a great two-solution problem here. We had things like macrocytic glycopeptides that came out, vancomycin, teicoplanin, work incredibly well for a sort of new diverse set of molecules that could not be addressed in those original columns that came out, and started to be more compatible with things like reverse phase mode and mass spec compatibility for the diverse compounds that we started to see. And then moving into prep scale, initially a lot of the particle sizes that were available were things like 10 micron or 5 micron. We also had, again, limitations in things like solvent compatibility. So in the early 2000s, we started to move into larger particle sizes. We have now 20 micron available, which is great for really large scale prep projects. We had innovations in technology. We had things like simulated moving bed chromatography that came out that allow us now to do metric scale chiral separations. We had the introduction of immobilization, which allowed us to now use things like dichloromethane and ethyl acetate. So overcoming solvent and mass spec compatibility, we had the immobilization, which was incredibly important. It allowed us to do things like overloading with the use of DCM, or this is a DCM THF mobile phase here, something that you would never ever want to use on AD. Now, because we have something like IA available, we can do a really effective prep separation of this compound. The ability to immobilize something like Crown Pack. There is a coated version of Crown Pack which is sensitive to a certain level of organic solvent. If we immobilize it, now not only can we go to a much higher level of organic, you're going here from 20% acetonitrile to 96% acetonitrile, but we can also switch our additives from something that was not mass spec friendly like perchloric acid to something that is more mass spec friendly like TFA. So all this sort of development and technology over time has led us to a pretty good number of solutions that are available now to us that solve a lot of diversity, a lot of problems with the diversity of chirality that we have that we're facing at the moment. But there's still obviously plenty of room to innovate. There's still plenty of new column options that are coming out, and certainly plenty of challenges that need to be solved as we continue to move forward. But let's jump in now and start really drilling down with the polysaccharides, because as I mentioned, Dicel Chiral Technologies, Dicel Corporation, these are the phases that we are most well known for. They are the ones that are the most widely used across the broadest diverse landscape of chiral molecules. So it's worth doing a little bit of a deep dive into these and discussing the separations mechanism. So that's what we'll do. But first, we'll start with the naming. So the naming of our columns could potentially be a little confusing. There are 47 of them, but the product names that you will see used most often are going to be this Chiral Pack product line name, They are given to all of our coded amylosic phases, as well as all of the immobilized columns that we have available. And then you're also going to see chiral cell, which is given only to the coded cellulosic phases. Now, how do we tell the difference between these product lines? It comes down to the first letter of the two-letter selector of the column. So anything that starts with an A is going to be a coded amylosic phase. Anything that starts with an O is going to be a coded cellulosic phase. And anything that starts with an I, whether it's amylosic or cellulosic, is going to be an immobilized column. Now, the difference is there has been a physical covalent linkage installed in the immobilized phases to prevent things like polymer degradation or polymer removal by exposure to dichloromethane or ethyl acetate. So while some of these selectors might have the same structure, the immobilized columns have compatibility with what I'm saying here are forbidden normal phase solvents or extended range mode solvents like DCM that would be destructive to our coded phases. So we want to stay away from the use of those solvents with these coded columns whenever possible. Now, the specialty selectors, we won't talk about them in detail, at least from a separations mechanism standpoint. We have touched on them, though, in some of those earlier slides with the development of these phases. The chiral pack name is oftentimes associated with those, but you'll also see crown pack associated specifically with the chiral crown ether columns. But here's your ligand exchange, here's your protein columns, here's those specialty selectors coming from Wolfgang Lindner for anion and Zwitter ionic molecules. So Dicel has quite a diverse stable of chiral columns to address a lot of the different unique needs that come up in chiral chromatography. Of all the chiral selectors that we have available, there are 47 in total. There are 37 unique selectors. That's because there are some that are shared between the coded and the immobilized technology. So as an example, IA and AD both have this 3,5-dimethylphenyl carbamate on amylose. However, IA is the immobilized equivalent and AD is the coded equivalent. So we do treat them as if they are separate columns because of the expanded solvent range compatibility. We can do some different things with IA than we could with AD, and the selectivity is oftentimes a little bit different because of that immobilization process. So for all intents and purposes, we have 47 columns. The selector types are 37. And then in some instances, you've got the same chiral selector available both as an amylosic and a cellulosic. So again, sticking to the top of the slide here, you've got IA and IBN next to each other, both 3,5-dimethylphenyl carbamate, but IA is amylosic and IBN is cellulosic. And that will almost always give very different performance in a chiral separation. So having access to both of those polymers with that selector is often a very helpful tool for us as we begin trying to develop and optimize our chiral separations with the polysaccharides. So talked about separations mechanism for achiral, it's mostly polarity. Let's talk about what it looks like for polysaccharides. There are 6 conditions that need to be met in order for us to see a chiral separation. So you might be thinking to yourself, 6 sounds like a lot more than just 1 with polarity, and in fact it is. And this is part of the reason why there hasn't been a very good predictive software launched yet for developing chiral separations, at least not a foolproof one where you get an exact answer of this is the column, this is the mobile phase that you should use. There are 6 different things that we need to account for when we're talking about a chiral separation and whether or not it can happen. The first one is the formation of transient diastereomeric complexes, which is really just a fancy way of saying that the analyte, the chiral molecules, have to be able to interact with the chiral stationary phase. They have to interact long enough with the chiral selector that you actually form this complex, but not so fast that we don't see the energy difference that's taking place, but not so long that it is essentially an irreversible complex that forms. So that's to interact, but not too long. It interacts in a series of intermolecular interactions, which I have shown in this table here. Hydrogen bonding being one of the most important ones that we'll discuss, but also things like steric hindrance, pi stacking, dipole-dipole interactions. And so when we talk about these enantiomers, if they bind essentially with the same geometry and same energy, then we're not going to see any separation take place. You might have seen this represented previously or in other presentations as the 3-point interaction model. In reality, there's probably far more than 3 interactions taking place at any one time, but trying to represent 3-dimensional space in 2-dimensional slides, it's much easier to draw it just with 3 points of interaction. But the idea here is that you've got a chiral selector with 3 points of functionality. You've got your pairs of enantiomers, which can come in and form these diastereomeric complexes. One of them here on the left-hand side has a better alignment of the functionality with the chiral selector. So you have a stronger energy of binding of that complex when it forms, higher energy, more retention, later elution. On the right-hand side, same functionality, non-superimposable mirror images though, so oriented differently in space. The energy of that binding is different, therefore lower energy, less retention, earlier elution. The second condition that needs to be met is there has to be a difference in the binding energy of these two complexes when they form, represented by double delta G or Gibbs free energy. So a separation is only possible if the two enantiomer selector complexes that form have different stabilities or a different energy. When we're talking about just one of these complexes forming, and we want to look at how does that translate to all of these chromatographic factors that we talked about earlier in the slides, the total Gibbs free energy of one enantiomer interacting with one selector directly relates to the retention factor. So the longer or the higher the energy of binding is, the larger the retention factor is going to be, the longer that thing is going to be retained on the column. But as we start to talk about the differences between the two complexes themselves, the larger the difference between the two complexes directly translates to selectivity. So I talked about the two handles in chromatography that we have a hold on, retention factor and selectivity. Now we're connecting it to these complexes forming and the energy difference between those when they form on the column. We only need a 0.2 kcal per mole difference though between these two complexes in order to see an observable chiral separation. So what does that mean in context of these intermolecular interactions that can take place on the column? Let's take hydrogen bonding as an example. The typical interaction only accounts for 1 to 5 kcals per mole. So depending on how you're looking at it within that range, we're talking about affecting one hydrogen bond 5 to 20% in order to go potentially from no separation to a separation, or to go from a separation to no separation. So we're not talking about very large changes required in order to massively affect our chiral separations. If we want to look at it from a binding curve example here, you've got a complex A in blue and a complex B in red. Out on the very fringes or the very far edges of this binding curve, you're going to have a double delta G Gibbs free energy difference that is much greater than 0.2 kcals per mole. So you're going to see very well resolved peaks. And of course, because they are well resolved from each other, a very good selectivity. Now, as we start moving closer to this point of intersection, as long as we are 0.2 kcals per mole or greater, we will see baseline or maybe partial coelution, depending on where we fall in this region of the curve. But again, selectivity should be reasonable. As we hit this exact point of crossover where there's now no difference in binding energy at all, of course, now we are going to have coelution completely, no selectivity whatsoever, no separation. Condition number 3, it's not enough to just talk about the energy and having a difference in energy. The actual chiral stationary phases themselves have to be positioned in the right orientation and be flexible enough to account for or accommodate these chiral molecules in order for us to see a separation take place. So what Professor Okamoto and others in his group showed is that these chiral stationary phases based on cellulose and amylose form left-handed helical structures, and in doing so form a repeating pattern of grooves and pendants. So you'll see here this 4-leaf clover pattern. You've got these little wedges on the outside corners of the chiral stationary phase here, which is what we would call our chiral grooves. And along the outside edge of those pendant of the chiral grooves are your pendants in all variety of different orientations. And those orientations and the overall shape and size of these chiral grooves is going to change depending on what the mobile phase is. So when we start to add things like alcohols, it's not going to be surprising if we see the overall chiral separation shift, or in some cases, even see things like reversals of elution order, just by changing the mobile phase, same column, same analytes. So whether a separation can occur depends very much so on whether the selector conformation available in a given mobile phase is the exact right conformation in order to allow for discrimination of these enantiomers that are being exposed to the column. So you can see already, we're only up to condition number 3. There's a lot of stuff going on this column, which is, again, partly why it's really challenging to predict which column is going to work. Cellulose versus amylose. When we look at the overall shape and size of these polymers and we compare them to each other, on the left-hand side, you've got chiral cell OC, which is trisphenyl carbamate on cellulose. On the right-hand side, the amylosic equivalent. The overall shape and size of these, although they have the same chiral selector, is very, very different. So if you know nothing about chiral chromatography, just visually looking at this, I'm sure you're assuming that these will perform differently in a chiral separation, and in fact, almost always they do. We can talk about the actual shape and size of the chiral groove, though, as it relates to the pendant structure. So when we have things like 3,5-dimethyl, which is our AD and OD type chiral selectors, we see more of this cup-shaped cavity where there's a lot of good access for a broad range of sizes of molecules, which is partly why AD and OD are still to this day 2 of the most popular phases that are sold and used for chiral separations. But we look at something like 4-methyl, which is chiral cell OJ, while we have really good access at the very opening of the pocket, it starts to narrow as we get into the core of that polymer. So we start to see a little bit of a restriction and maybe some size exclusion that can take place because of the shape of that binding pocket. Then you have something like 2,5-dimethyl, where it's more of a hindered cavity, which means that your shape has to kind of approach on a specific angle in order for entry into that binding groove. So chiral conformation flexibility plays a large role in an ability for a separation to take place, not only because of the solvent, but the shape of the chiral groove changing as well. Condition number 4, we know that certain molecular features, specific functional groups, increase the likelihood of seeing a separation. So one of the biggest drivers for our separations mechanism is hydrogen bonding. So the more donor and acceptors we have, oftentimes we see better success rate with those molecules separating on our polysaccharide phases. More aromatic rings means that we have more opportunity to pi stack. So more aromatic rings oftentimes increases the likelihood of seeing a separation, especially when they are located near to or in proximity to the chiral center. Bulky substituents, again, also when they are located near a chiral center, can, in some cases, fit nice and snug into those chiral grooves, meaning we'll see really nice recognition and really good separation. Or if they're too big, we start to see a clash and a loss of binding. So bulky substituents can be helpful or hurtful depending on the overall shape and size of that chiral selector. And rigid stereocenters are oftentimes better than having floppy extended chiral separation or chiral molecules. Floppy molecules, things that are, let's say, long alkyl linear structures where the chiral center is way far out on the end. There's a really large degree of rotation. And so while they might interact with the chiral stationary phase, oftentimes the energy difference averages out and we lose the selectivity or we lose the separation. Whereas we have rigid stereocenters that are located near either fixed structures like aromatic groups or in a ring structure like this, there's not a lot of ability for that stereocenter to rotate or get out of the way. And so we see better separation, better discrimination with those types of molecules. Condition number 5, the mobile phase environment. We talked about how things like alcohols can change the shape and size of our chiral grooves. It can also result in things like reversals of elution order, but the mobile phase can also compete for binding sites. So when we have things like a lot of methanol or a lot of water around, they can come in and actually bind or block some of those hydrogen bonding sites that would have otherwise been helpful for promoting a chiral separation. So when we're looking at things like normal phase mode, where we have hexane ethanol, hexane isopropanol, we're oftentimes seeing more hydrogen bonding activity going on. So the addition of water or the addition of methanol can really play around with normal phase separations. Reverse phase mode, we start to shift a little bit more towards hydrophobic and pi stacking interactions, so we can shift our mechanism depending on the solvents that we have present. So we can look at it something like this, where you've got points of functionality on our chiral selector that are solvated by our mobile phase. You've got points of functionality on our chiral molecules, which are also solvated. In some mobile phases, you might have complete solvent exclusion, and you have a nice tight binding of that diastereomeric complex. But in other cases, some things might remain solvated and block some of those interactions that would have otherwise taken place. So if you've ever tried to move a separation from, let's say, reverse phase mode to normal phase mode, or SFC mode to normal phase mode, you might notice some differences that are taking place and some of that has to do with this binding sites being blocked by the mobile phase. And like I mentioned, changing the shape and size of the chiral grooves can reverse the elution order, but it can also do it by competing with the binding sites as well. So here you go from a 100% methanol with your positive enantiomer eluting first on chiral cell OJ. We begin adding ethanol to this separation to the point where we switch over to now 100% ethanol, and we reversed our elution order just by changing the mobile phase. And last but certainly not least, because all 5 of these other conditions are not complicated or complex enough, we have to make some considerations about kinetics and thermodynamics. It's not enough to just have an energy difference of 0.2 kcal per mole, and then we see a separation. The exchange rate between free and bound state of these diastereomeric complexes has to be in the right window. The exchange can't be too fast, because then again, the complexes end up averaging out and we don't see a separation. They can't be too slow, because then the peaks end up broadening out too far and we end up losing separations that way. So there's this sweet spot, sort of like Goldilocks and the 3 bears. The porridge can't be too hot, can't be too cold. It has to be just in the right window, the right temperature, in order for us to take advantage of all 5 of these other things that we talked about. So if the exchange is too fast, too rapid, yes, there could be an energy difference to where you theoretically should see a separation, but everything averages out and you see no separation at all. Here you've got a nice slow oscillating on-off of those complexes, a nice exchange, a nice baseline separation here. If the exchange is too slow, here the peaks broaden out too far. Obviously, if the exchange was faster, these probably would be 2 nice resolved peaks from each other, but because now they've broadened out so far, we don't see the separation anymore. So 6 conditions. There have been publications done that have modeled some of these interactions and have come up with models for specific cases, but trying to make it universal, trying to make it apply for sort of any sort of new entity that comes in has not been done yet. And so one of the reasons why we harp on or advertise or advocate for a screening process is because that is still one of the most efficient ways for us to find chiral separations on the polysaccharide chiral stationary phases, and really on any chiral phase for that matter. Here, linazolid, screening it on IA through IN, we see that IG happens to give us a really nice separation of the 2 enantiomers. And it also gives us an opportunity to start answering questions about solvent effects for things like reversal of elution order. You notice here when I made the sample up, I didn't make the sample up 1-to-1. The separation here on IBN shows that the second enantiomer is in less concentration than the first enantiomer. Then we go up here on IF, we notice that there is a reversal of elution order here. So we can start answering some of these complex questions just by taking it through a screening process. And I will be honest with you, if optimized correctly, your screening can be done in a few hours as opposed to a few days or even weeks to try to do the computational modeling to answer the question of which column you should be using. Now, the second to last section here, applications and examples. Like I said in the very beginning of this presentation, there are simply far too many examples to try to go through in a short period of time, like an hour. So I'm just going to show you some major classes of separations, perhaps to be inspirational for you to see the broad utility of these columns. But if you have specific application questions or you're interested in learning more about these specific things, in the closing remarks section, I'll show you where you can find out a little bit more information, things that we've already worked on and developed, but also email addresses that you can reach out for us to be able to have that conversation for specific things that you're trying to work on. Of course, small molecule chiral compounds have been where these columns have been used for literally decades now. Just a few examples here, you've got something like naproxen, the small chiral molecule separating well on IK. Under normal phase conditions here, amlodipine on IM, we're starting to broaden out here a little bit in our additive range. We've got ethanolamine, which is a little bit of a non-traditional additive, and then things like protected amino acids here on IN in SFC mode. So a lot of diversity, a lot of broad applicability with small chiral molecules. But our chiral columns are not limited to just doing separations of chiral molecules. They can also be used for what I have noted here as mixed mode separations, or mixtures of enantiomers and diastereomers, or enantiomers with structural isomers. So here you've got an example of cannabicyclol, which is itself chiral, and cannabichromene, which is also chiral, but comparing them to each other, no relation. You're doing a mixed mode separation here, nice 4 peaks resolved. Same thing here with THC isomers. You've got delta 8 plus and minus, delta 9 plus and minus. They are enantiomers to each other, plus minus 8, plus minus 9, but not related to each other in terms of being enantiomers between 8 and 9. All 4 peaks resolved very nicely with ChiralPack IF in this case. Atrope isomers, I talked about those. You're talking now about a rotational axis of symmetry as opposed to a traditional chiral center. Your axis of rotation is located in cetorazib here between this CN bond. You've got 2 different conformations of the isopropyl group being either here on the outside or on the inside, separating here nicely on IK or on IE using 100% acetonitrile. So not just separating traditional chiral pairs, but also things like atrope isomers. Constitutional isomers, here we've got fluorofentanyl. We're moving the fluorine around this phenyl group, and we can do separations nicely here using ChiralPack IK in normal phase mode of all 3 of these constitutional isomers. Things like amino acids. I talked already about Zwick's earlier. Zwick's is one of those specialty phases that was developed by Wolfgang Lindner for separation of amphoteric and zwitterionic molecules. The nice thing about this one is there is some predictability in the elution order. At least for amino acids, you'll always notice that Zwick's plus elutes the L amino acid first. And if you need to reverse the elution order, you go to Zwick's minus, and the D amino acid comes out first. So these are nice columns to use in these separations. And as we start adding complexity to our molecules, things like oligonucleotides or peptides also separate very well on our chiral columns. Oligonucleotides and amino acids are still a bit of a challenging separation for us. Amino acids require oftentimes a few different columns in order to do separations. We're very honored to say that tomorrow we have a new column that's being launched specifically for amino acid analysis. So keeping in mind, we have a lot of people here from a lot of different regions around the world. If this is a column that sounds like it's interesting to you, I would encourage you to reach out to us for more information, and we would be happy to have that conversation with you or point you to the right Dicel affiliate to have that conversation with. Closing remarks. As we close in here, we've got a few minutes left, just some helpful information for you. As we leave today, our website, chiraltech.com, has a lot of really great information, especially under this service and support tab. You will find things like our technical application database, our method development guides for all of our columns that are available, help you set up screening. We have a link there to all of our column instruction manuals as well, which is incredibly important. We have a YouTube page. You can go search for us, Dicel Chiral Technologies. We have a number of dedicated webinars for things like method development, which is probably the next step from today. If you want to learn more about chiral chromatography, the next step is learning about method development. So we have a webinar there that addresses those topics, but also things like method validation and optimization, sub-2 micron chiral phases that we have available. So definitely check out our YouTube page and get some more information there. Under that service and support tab, you'll also find our Global Application Center, which is a series of generic pharmaceutical compounds that have been screened and methods validated by our group in India, Dicel Chiral Technologies India. You can go there, click on those links. You get some really nice chromatograms that give you the starting conditions for screening, the column that's used, and then the relevant chromatograms associated with those separations. Specific here to the US, and I think most of our Dicel affiliates around the world also offer this service, but when you reach out to them, we can clarify that. But here in the US, if all of this talk about screening and all these different interactions is a bit overwhelming, we do offer a free column screening service for analytical separations. So you can reach out to one of the salespeople who are on the last slide. I'll show you their email addresses. You reach out to them, you can send us a sample, we'll screen it for you for free, give you a chromatogram, separation conditions, and a no obligation quote for that column to buy and then reproduce that separation yourself in your own lab. And then if you want us to keep working on it, keep developing, keep optimizing, we also have a for-fee service that you can take advantage of where we'll keep working on those separations and get you something that's hopefully closer to your expectation for optimization. And then last but certainly not least, some email addresses. Again, there are some regional separation that needs to be acknowledged at this point. Chiral Technologies, Dicel Chiral Technologies, which is who I am a representative for, covers North and South America, either directly or through a network of really awesome distributors. I would encourage you to reach out to myself, Austin Bailey, or Tracy Hartledge if you have any questions about column sales or sample submission. We might directly handle those, or we might send you to one of those distributor partners here in North or Central South America. We also have Dicel affiliates in France, which cover Europe, Africa, parts of the Middle East, DCTC, which is Dicel China, covering China, India, Japan. We will direct you to where you need to go. So if you're not talking to us, we will make sure that you get to the right person. But here are the email addresses for you to reach out to. We will take care of that for you. Any questions that you have, we still have some time. I will pass it over back to Austin now to do a nice little outro. But again, thank you very much for being in attendance today. And hopefully you can appreciate a little bit better chiral chromatography, our phases, and you'll be interested in continuing your journey, learning more about these with us. So Austin, back to you.
Thank you very much, Wes. We all certainly appreciate your knowledge, expertise, and excitement towards chiral chromatography. First off, I do want to start off with some of the questions that we have in the chat. The first question that we have is from Julie. For the column ChiroPack ADH using 100% ethanol, can we use IPA or methanol? What is the principle for choosing mobile phases?
I should probably unmute myself while I'm answering the question. ADH, because it is a coated phase, does have some solvent restrictions. However, alcohols are not one of them. So you can use 100% isopropanol, you can use 100% methanol. The only issue really with using something like 100% ethanol or isopropanol is because they are much more viscous, you have to be more mindful of column pressure. So I would encourage you to reference the instruction manuals to get a lot of details around how to adjust flow rate, how to accommodate things like column pressure. The coated 5-micron particles like ADH can go up to 300 bars, so there's usually pretty good flexibility in how you can use them, but you might find that you have to reduce the flow rate on those columns to 0.5 ml a minute. Principally though, which mobile phase you use is going to depend very heavily on all these other 6 parameters that we talked about. In polar organic mode, which is methanol, ethanol, isopropanol, generally speaking, as we go from methanol to isopropanol, we should see an increase in retention because isopropanol is less polar than methanol. We should see a natural increase in the retention. However, these interactions are very complex, so it is possible that methanol might actually give you more retention than isopropanol. So there are some predictabilities, there are some unpredictabilities. Screening is oftentimes the best way, again, to answer the question, though, of which alcohol you should be using or which mobile phase you should be using.
Great, thank you so much, Wes. Next, we have a question from Aritra saying, can you please suggest some columns for rotamers or positional isomers separation in both SFC and normal phase mode? Now, I'll go ahead and say more than likely we would defer to our free screening service to make that recommendation, but I'll let Wes address that here.
Yes, definitely. I did show an example in the applications there of the, and I still am sharing my screen, so I'll go back to them, the positional isomers of fluorofentanyl. There is not one chiral column that works better for rotamers or positional isomers. The approach for these is oftentimes, again, screening and finding which ones work best. We do have a number of achiral phases, which obviously I didn't talk about that in this presentation because we're focused on chirality here, but we do have a number of achiral phases that are good for separating positional and rotational isomers. PBT, P4VP are 2 of those. If you want more information on those, you can reach out to the email addresses and either one of our sales reps or someone in your region can reach out and give you more information on those. PBT in particular has been shown to work pretty well for positional isomers, like I have shown for the fluorofentanyl isomers here, But the chiral phases, it is a screening approach, and I can say that I have examples on most of the phases where you can do those separations.
All right, thank you, Wes. And I do have one here from Mehmet, just asking, am I hearing you right, that you will announce a new column for separation of amino acids? Yes, that is correct. We are going to be releasing a new column here this week. I will go ahead and just let Wes, add any more that he feels necessary to that.
Yep. I'm trying to keep the details a little bit hidden only because, again, some regional differences and dicel affiliates around the world, we're all working together on this, but I don't want to, I want them to be the ones that are sharing all the good news with you. So if you're located in our market, again, North, Central, or South America, reach out to any one of these email addresses and we can give you some more information or direct you to the dicel affiliate that will handle that. But it's a brand new column that's going to be capable of separating 21 proteinogenic, naturally occurring amino acids, both chirally and achirally, in 5 minutes. So we think it's going to be pretty revolutionary, and I'm not saying that just because I am an employee of the company, but it is a pretty monumental development for the field, especially for amino acid separation. So reach out to us, and we'd love to have a more in-depth conversation with you to give you all those details.
Thank you, Wes. So next we have a question from Francis, asking, how do you resolve peaks of enantiomers when one is in the positive mode and the other is in negative mode on ChiralPack ODH?
I'm not sure what you mean by in the positive mode or in the negative mode. I don't know if that person is still here, if they can clarify. When I hear positive and negative mode, I'm thinking mass spec, or it could also be mixed mode functionality, but I'm not quite sure.
Yeah. And Francis, if you can clarify, let's see. Francis did clarify here saying one facing upward and the other downwards.
Okay, so sort of like this binol or naphthol type compounds. So back to the original question. Can you read what the original question is also? How do you separate them?
Yes. How do you resolve peaks of enantiomers when one is in the positive and the other in the negative for ODH?
Gotcha. It would be the same approach. Now, I will say, because you're asking specifically about ODH, there really aren't any foolproof ways of saying, this is how you would separate them. Normally, when we're, again, given these sort of which column should I use for this compound, we're going to be screening it. It is very possible that ODH could be the one that's giving the separation, but it's hard to say for sure that OD would be the one. So again, it's sort of a lame answer, but it is the answer that usually gives the best result, which is we have to screen it. If you want to just stay with ODH, I would encourage you to look at our method development guide. And there's a number of screening protocols that are in there. If you just want to stick with ODH, how you can kind of manipulate the mobile phase landscape around what OD is compatible with. And you might find that maybe going from SFC mode to normal phase mode or normal phase mode to polar organic mode allows for a separation on ODH. But again, it's still an unpredictable modification there of mobile phase to hopefully elicit or extract some sort of a separation.
All right. Thank you very much, Wes. All right. So next we do have a question from Paul. Paul is asking, since the ADH and the IA have the same chiral selector, will the separation be the equivalent under the same conditions?
No, and it's not because they're somehow different from each other or one's deficient or whatever. The immobilization process, the actual installation of a covalent linkage between the silica and the polymer, does restrict some of the polymer's ability to flex and accommodate the analytes when they are introduced to the column. So going back to, I think it was condition number 3, where we talked about selector conformation and polymer flexibility, the restriction of the immobilization process oftentimes means that we see slightly less selectivity in the case of an immobilized column versus the coated equivalent. However, because of the access to extended range solvents, we oftentimes can find better separations by incorporating some of those restricted solvents with redeveloping the method on IA, as an example here with going from ADH to IA. So yes, there are differences. Usually it's a slight decrease in the selectivity, but we have ways to modify it to improve it actually over what you might be seeing with ADH.
All right. Thank you very much, Wes. And we did have a question from Stephanie Collingwood, which was answered in this previous question here. So,
Okay.
Stephanie, thank you for asking, and I'm glad that we could get you an answer here. And I do want to say there are a few questions that are more case specific. I have a few more general questions that I'll get through, and then we'll get to some of those specific situations here. Now, actually, we will get to those specific questions right now. So we have Stephanie Becker that's asking, saying that she is working on a dipeptide, FMOX starting materials that contain both L and D isomers and their corresponding allo forms. I've already tested amylose, cellulose, and QNAX-based chiral columns, but I'm still seeing co-elution. Do you have any other column chemistry or stationary phase recommendations that might better resolve these stereoisomers? And that question has been forwarded to you, Wes, so you can see the entirety of the question as well.
Okay, let me pull that up then and check it out here. Well, the first thing I heard was allo, which I'm assuming is probably leaning towards allo-isoleucine, which is a very challenging separation. I will say that our brand new column that's coming out tomorrow has applications for certain mixtures that you've mentioned here in this case. Dipeptides, we have found that dipeptides work even up to tri and 6-mers, do work on Zwix, which is the zwitterionic amphoteric chiral phase. So it's possible that Zwix might be a little bit of a better option, but I would be interested in having you try our new column because I have a feeling that it could work quite well for what exactly it is that you're trying to do here. So I would encourage you to reach out to us so that we can give you some more information on how to get that.
All right. Thank you very much, Wes. Now we do have another question here. Excuse me 1 second. A question from, I'll get to it here in a second. Ivan, good to have you here. This is a more specific question. I've just forwarded it to you, Wes, so you can see the entire question. He is saying we currently purchased a 4mm by 100mm 5-micron L108 packing chiral pack CBH column with the column guard, using to analyze their final product, which is 2% lidocaine and 1 to 200,000 epinephrine, mostly water and salt, pH right around 4, and our raw material API epinephrine bitartrate. We are following a USP method, epinephrine bitartrate. Our mobile phase is 90% monobasic sodium phosphate in water, EDTA, disodium salt, and 5% IPA, pH right around 5.8. Would this column be able to detect 5 micrograms per milliliter of epinephrine at 280 nanometers? Also, in your experience, have other customers use this column to detect epinephrine, and what would the lowest limit of detection theoretically be?
So this is not something here that this question is not specifically about our columns per se. It's more about the detection of the actual analytes. So my assumption is that the detection method is probably UV based. I'm almost positive that monobasic sodium phosphate is not mass spec compatible, or at least is not a great option for mass spec compatibility. So when we start talking about really low concentrations, at micrograms per mil, Usually it's still in the range where we can detect it pretty reliably by UV. However, a lot of it does have to do with the UV signature of the actual analytes themselves and what else is around it. Is there any sort of competing impurities that are causing issues with quantification? I can't say that I've worked with this method directly to be able to say for sure that you should be able to detect or quantify 5 micrograms per mil of epinephrine. I could say for sure that probably if you had the ability to check mass spec, that you would definitely be able to do that. But UV based, I unfortunately can't give you an example or a direct experiential answer. Lowest limit of detection, theoretically speaking, yeah, for this specific method, I have not talked to anyone specifically about this method to know what they have done. I would like to think that if it is a USP validated method, that it should be okay, but I can't say for sure.
Thank you very much, Wes. And that's all for the questions. We do have, so I do have Mehmet wrote and just asked for some information on the amino acid column. Mehmet, I'm glad that you can make it here, and I'll be sure to send you information as soon as we have that available for you. And yeah, that is all. That's it for all of our questions here today. We are past that time, the hour that we have for today, so we will hop off here. But again, I want to thank you all for joining in. And any other questions, we'll be more than happy to answer those separate from this webinar. And Wes, thank you again for your time and your expertise. And everyone, you have a great rest of your day.
Thank you, everyone.